So the first speaker for you today is Anne Graybiel. Anne is one of MIT's most distinguished faculty. She is institute professor. Only 12 faculty out of nearly 1,000 at MIT hold this honor. She is a member of the National Academy of Sciences, of the Institute of Medicine. She has been awarded the National Medal of Science, this nation's highest scientific honor. Anne is the world's leading expert on the basal ganglia and on circuits that connect this large forebrain structure to the cerebral cortex. Anne's work demonstrates the involvement of the basal ganglia in the control of movements, in the formation of habits. In fact, given the central phenotype of repetitive restricted behaviors in autism, it is amazing how underappreciated the role of the basal ganglia is. But Anne's work will change this. Anne.
That's extraordinary. I think you've heard the best already. So it's been a wonderful day. Thank you so much for having me. I vividly remember going to the early meetings when this autism consortium was just beginning. Everyone was so hopeful something good would come of it, and we all gathered, or I say all, there weren't very many of us, in a little tiny room, and it was kind of crowded, and we talked with one another, and look what's happened. It's just amazing. Now, we've heard many things today, and we've heard about the extraordinary advances in the genetics that could be related to autism spectrum disorders. And I think we're all in awe of what can come from that. And I just want to return to those core symptoms, the reason that the family members have come today, those core problems that we face in one way or another in engaging with someone with autism and trying to help. And Riganka already said it, we're interested in brain circuits, whole networks of neurons in the brain that communicate with one another, and as you just heard before the break, these communication lines can become disabled. And so we have been very inspired by this field. Our lab has joined it in gusto, and I want to tell you a little bit about that. I want to tell you two stories that we think may interest you, where I especially want to interest the family members here. You're so important to research. So the research that I want to interest you in deals with the basal ganglia, and the basal ganglia are deep lying in our brain underneath that outer rind of cortex that Riganka talked about. They still are rather mysterious parts of the brain, except neurologists knew for a whole long time that somehow the basal ganglia, or extrapyramidal motor system, somehow that system is important for disorders that used to be called motor disorders and now are called motor plus disorders. Why? Because in many of the disorders that fall in this category, including Parkinson's disease, there are emotional signs or cognitive problems that go along with the motor disabilities. And I think that was a very early sign for what might come, and what has come is that now we know these wonderful basal ganglia, which used to be thought simply to release and inhibit movement, to get a lot of input from the neocortex shown in this little cartoon running down toward the basal ganglia. We now know that these regions of the brain are also implicated in a number of neuropsychiatric disorders. And I've had the audacity to put autism here, but I want to show you it's not just audacity, because Tourette, OCD, autism, ADHD, and for the sake of space, if not time, I've not included some others, now are in the basal ganglia family of disorders. And these now, interestingly, often show comorbidity. It's called, so that, for example, as I'm sure everyone here knows, autistic spectrum disorder, ASD problems, co-occur a lot with OCD, and I'll come to that, because we now think, many in the field and in our lab in particular, that maybe there's a whole cluster of disorders related to these basal ganglia circuits as a fundamental problem, and we hope that maybe these circuits, which we know to be related to just normal habits and routines, we hope that by understanding these circuits and then manipulating them, we can help in the field of autism. So there's something very special that's happened quite lately, and that is that Guoping Feng and his collaborators have managed to find 2 genes, one, the autism gene SHANK3, the other, the SAPAP3 obsessive-compulsive disorder gene. Both of these turn out to be concentrated very, very significantly in the basal ganglia, in particular in the striatum, this little round ball that I keep showing you in a cartoon. That is terribly important because we know then we have a big clue that maybe genes that are heavily expressed in this region and are related to autism, we might use them as clues for what's going on. And in fact, if we go back to those main problems with communication and language, now we know the FOXP2 gene, the famous human language gene, is also shockingly heavily concentrated in the striatum, not in this neocortex so much as in the striatum. And you can see then that for each of these behavioral problems, there are now known genes that are striatum enriched, deep in those basal ganglia zone I showed you in the cartoon, and they have something to do with autism. So I'm going to concentrate, we are working very much on the FOXP2 language gene. I'd love to show you those data, but I'm going to concentrate, as Mriganka said, on these repetitive disorders because I know, you know better than I know, but I know from talking with many people who have autism in their families that these are some of the really tough problems to deal with. The tendency to do something over and over, or, as I will come back to be just especially focused, too focused on one thing, stuck. So how could it be that the basal ganglia, I just admitted they're deep in the core of the brain, how could they be so important for these behaviors? And part of the answer seems to be that the basal ganglia have massive physical connections through synaptic roots up to the very front end of the neocortex, the part that we think of as being very, very important for high-level behaviors, certainly including language and social behavior. And so the upshot of all of this is we have a picture of these circuits. It turns out that they're kind of like loops between the neocortex, the striatum, back. They're influenced by the famous dopamine, important in Parkinson's disease, as it goes down. And the amazing thing is that this modulating pathway seems to have something to do with reward or reward prediction errors. And this has been meaning that probably it's basically the striatum's a happy place. For example, it lights up when people in MRI get to see pictures of the chocolate they're about to get. I would imagine that just looking at this, your striatums are glowing. Mine definitely is. But what happens is we've got a system then in our brains that helps us learn. It gives us a kind of reward after certain behaviors. And so with this system, the loops going round and around, we develop habits. Now, what happens if the loops go on and on too much, so we sort of get too much of a habit? could it be that some of the disorders that we see when we think of the very repetitive behaviors and very focused behaviors of some kids in the autism spectrum series, could that have something to do with their behavior? So my two stories in brief are this. We hit on this many years ago when we gave a drug to rats. The drug makes them a little bit hyper. And in those days, we couldn't do, well, we still can't for these deep brain regions, a lot of imaging. But back then, the best we could do was to look at what genes were turned on in the brain very rapidly after that. And we took slices of a brain of a rat who had been given one of these psychomotor stimulants, and lo and behold, we saw these clusters of neurons. They're shown in white here. And it was then clear that, gee, if you give a little shot of a psychomotor stimulant to an animal, that these little clusters in the striatum light up. And when I say light up, I mean genes are turned on, and these genes are transcription factors that control how other genes turn on and off. And the story got interesting because very good behaviorists came to the lab for a postdoc, and what we found was that the more that pattern got patchy, the more the animals were repetitive in their behaviors. And we did this for a whole series of drugs with wonderful people who came to the lab. We then did it with monkeys. Here is a monkey given one single shot of a drug. 2 weeks later, if another monkey has had 2 weeks of a psychomotor stimulant, the genes are turned on again in this patchy fashion. Those zones stand out, and moreover, here's the monkey chart. The more those patchy regions, we call them striosomes, are activated to express genes, the more the animals are stereotypic. Or put the other way, could this somehow tell us there's some kind of circuit in there that's related to repetitive behavior? Well, we knew what those little star clusters were because we'd found them some time before in human brain. So this is a slice of a human brain, and this is his striatum, and you can see these pale zones here. Those are all what we ended up calling striatal bodies or striosomes. They are neurochemically, they are neurotransmitter, awesomely highly specified and different from all the other part of the striatum lying around them, darkly stained here. Well, that was interesting to find the activity in highly repetitively behaving animals lighting up that system. And part of the reason it was interesting is in another old study, we had found that if we looked in monkeys, and these are cartoons of the medial part and the bottom part of the brain of a monkey, if we looked and said, well, what part of the cortex is wired in with these zones in the monkey, there were only two regions heavily connected. They're shown in red. And of all things, these regions were well known clinically in humans to be part of what people called the OCD circuit. Now, this got more and more intriguing, and in essence, what seemed to be coming out was that there are evaluative circuits in our forebrain that help us decide things are good or bad or evil or wonderful and so on, or cause anxiety or depression. And some of these circuits feed into these patchy-like zones, striosomes, which are the only striatal neurons allowed to get down to the reward-related system, which in turn, guess what, modulates the loops. So you can imagine how a circuit in overdrive could lead to repetitive behaviors. So then we decided, naively, well, let's go get us some new striatal genes. At that time, I had no idea how to do this. I went back to school, and with Hiroaki Kawasaki, who later worked in David Hausman's lab, we found a bunch of genes. I'm showing you pictures of their distributions in the striatum. And one gene is shown here in orange, and then the other gene is in green. And you can see, here they are again, those patchy regions, the striosomes. And Jill Crittenden has done a very beautiful job of making knockouts and conditional knockouts of these genes. And I just want to point out, you just heard about guanine nucleotide exchange factors in the last talk. These guys, they like to listen to a second messenger, to a signaling molecule, and then they like to, on the other end of the gene, they have a little motif that lets them change how active the RAS superfamily molecules are, like RAS and RAP. Absolutely amazing to find this in brain. These are genes that are, these RAS and RAP, famous in cancer. Well, Jill, here's Jill Crittenden. Jill did a knockout of that orange gene. Now, we figured the reason for doing it is we knocked out the orange gene, the green gene, that is striosomes, ought to be more active, and maybe we could get some behavior out of the animal. So here's what the findings are. Now you're looking at a little diagram. It's a diagram made as though you're looking from an overhead camera, and the little mouse is in a box, and he's given one shot of amphetamine. And when a mouse gets that, the first thing he does, he runs around, explores. Now you do it every day, and then you again look at this image of the tracker data from the overhead camera. Little mouse is sitting in the corner doing one thing over and over again. Now in a normal, that's a control mouse, it took about 2 weeks to have that happen, to get that stereotype behavior that I showed you before that lights up the little star clusters. But in the knockout that Jill made of this, the orange gene, in fact, often just on the very first shot, the mice were sitting in the corner doing the same thing over and over again. And it's stunning when you see this. So that was very good, but nowadays it's possible to make what's called a conditional knockout. So the knockout, excuse me, experts, I'm just going to say it for the families, because I'm at about the same level. Usually, if you knock a gene out, it's out from the time of the egg, etc. on. But in a conditional knockout, you can make it so it only is deleted after birth. And so that's what Jill has done. And I thought you'd enjoy actually seeing pictures, maybe, of what brains look like, slices through mouse brains. So here's the control, and here's this wonderful conditional knockout. And you can see this dark stain. That's the stain for the orange gene. And on this side, you can see it just go away during development. And I put that in. It's, of course, a very detailed data. But the reason I put it in is that everyone in this room is interested in development. There's no one in this room who is not. And I'm showing you here that this striosomal system, the star cluster system, it has an amazing development. These are those dark spots, I hope you can see the patches there, are from an embryonic human. And over time, that system here that you see embryonically develops into this glorious system. Now, I want everyone in the audience to say these are beautiful. I know it's sort of like maybe not beautiful, but it's important. Anyway, with the conditional mouse, sure enough, when Jill and her colleagues and me put these into the activity monitor, here's the control, run around like mad, and here's a little knockout mouse just going back and forth, back and forth, back and forth, over and over, and it just took one shot to do that. Now, what we have found from the conditional knockouts that we are very interested in is that it's this focus thing. They'll be moving around, and all of a sudden they just stop, and they'll sniff for a while, and they'll just do it. And then after a while, it's as though they wake up and they go somewhere else, and they do it again. So in essence, here is my first story for you which is that one little gene, but this time I'm not just talking about a gene, it's a gene, but it's in a circuit. We know the circuit, we can study the circuit, so we can say if this circuit is disabled, then at least this part of the set of problems related to autism can occur, and I'm leaving out for lack of time the studies in which Eric Berger, who's sitting right there, has collaborated, showing that there is a learning deficit. Now, story number 2 will be short, but again, it relates to Eric, who's in the 1, 2, 3, I don't know, about 6 back in the middle section, looking embarrassed. Here is his name. Eric made the coolest task to study the OCD model mouse. And again, there is high, maybe up to 50% comorbidity of OCD with some forms of autism. And so he has his mice in a little box, and here's a mouse, and if you drop water on the nose of a mouse, the mouse will groom. Okay? So Eric drops water on the nose of the mouse, as I'll show you, and then he plays a tone before that to condition the mouse that when the tone comes on, pretty soon the water will come. And so you know perfectly well what the mice do. They start grooming to the tone. But let me show you what happens. Here are summary data for four control mice. These are not sick, but they're the controls for the OCD mouse. These are the trial numbers from the very first trial all the way. Look how many trials the poor little guys go through. Here's time. So the tone comes on here, and the water comes on with this turquoise line. And every dot you see now is when the guy started to groom. We're taking videos, and then the videos are scored. And you can see it's really cool. It's actually very cool. When it begins to learn, the mice, they groom, of course, when the droplet comes, but then they get this anticipation, they get conditioned, just like Pavlov's dogs. So they're grooming away, the minute the tone comes on. The trouble is down here in the time scale. It's a couple of seconds until the water drop comes. So if you sit there grooming for 2 seconds, you're wasting your time. So they basically mainly stop doing that, but they get very good at the timing so that, boy, when that water comes, they go. Now, I'm telling you this because the poor little OCD mice are going to be different. And look what they do. Here's a tone. Here's a water. And they learn, right? In fact, they may learn better than the normal mice, which would in itself be rather interesting to the people in this room. But look what happens. Whereas the normal mice gradually realize, hey, it really isn't worth grooming to the tone, the SAPAP3 mice just can't stop doing it. That tone comes on, and they have a little compulsion, and they go after it, and they groom. Now, we are very excited to see this, excited enough so Eric and I have prepared a video presentation for you. This is the beginning of training, and you're going to have to look hard. It's very hard for me to see here. But, over here I can see feet. These are bottom views of the mouse. Here's his little hat he's wearing, and the water is going to drip on his nose. It's a little harder to see. Here are his feet up here. And so you will see here pretty soon, there goes a tone. You have to look at this symbol, and frankly, not much happens. But now, can you see that they're moving their feet? I hope the lights are low enough. Eric, can you see it? Good, okay. Now, let's go to the middle of training. Middle of training, the guys have become conditioned. So, let's see. So they're sitting there. I hope you can see that they are grooming, and then even more so. And now the last. His feet are here. I have a lot of glare. I think his feet are here. You need to look here and watch. Watch when the tongue comes on. Whoops. Oh, wasn't that clever? There it goes. See this guy? He's grooming away. The other guy didn't groom at all, so that's the demo that you see is really true. It's become the lab favorite. Eric's the most popular guy in the lab now because well, not only does he have wonderful videos, but also it's just a fascinating phenomenon. And here it is. I'm almost at the end of the talk. I'm trying to not make us too late. Here is a graph showing the percent of grooming bouts in this task. Here are the days of learning, and here's the control guy going up. And then gives up because it doesn't do him any good to groom. And here is the poor SAP-app guy who has somehow, through learning, developed what we think of as the drive to do the repetitive behavior. Now, that's where the story stops, except that the story is going on. Eric's here, so he isn't doing the experiment. But we're actually doing more because we want to cure this. And one of the wonderful things about neuroscience now is we're developing methods that may let us actually intervene in ways that eventually will go to the clinic. So we've decided to start with this. It's easier than starting with a more complicated kind of one of the other problems in autism. And so what we're doing now is rigging up the mice. Here's a mouse. He wears a little head stage. We're recording through wires that are sitting in here and attached to 1 million computers outside that go down into his striatum and other places in the brain. And the wonderful thing now is that in this rig, here's a side view of it, we can actually extrude a small little light fiber, that little light guide that goes down into the brain, and we can shine light where we have previously put via viruses, put a little light-sensitive molecule into just certain neurons. That sounds like magic. If you haven't heard about it before, this is a very new method developed at Stanford and by some people at MIT. 3 people at MIT were in the early stages of this. And so what we're trying to do now is see whether we can find the hot spot in the circuit I talked about with you. Similarly, the same circuit we think is the Jill mouse circuit that made them very stereotypic. Try to go in there and find a hot spot where if we shine the light to turn off the circuit, we turn off the behavior that is a bad behavior to have. So with that, I want to just say we're in the basic research side of things tremendously in awe of you guys for what you're doing to support the doctors and clinicians and basic scientists in every way. I wish the families good luck, and I want to thank the people in the lab who have worked so hard, Jill and her wonderful people who are working with her, and Eric and other wonderful people. Here's Go Ping Feng, and Safari and the Simons and NIH for their support. Thank you.